Hi, I'm Lauren. I'm a junior integrated science and technology major here at JMU.
Hi, I'm Grace Taylor. I'm a biotech freshman major.
Hi, I'm Jessica Garth, and I'm a biotechnology major, and I'm a senior.
The story of Bob begins in 2012 when one of the JMU viral discovery sections used a soil bacterium, Bacillus pumilus, as the host for phage discovery. Students have used B. pumilus for several years, having isolated it from soil and discovering dozens of interesting phages. At the time, B. pumilus was most well known as the bacteria most likely to survive the cleaning of spaceships due to its tough spores. Hoping to find more variety of phages, we obtained 2 more isolates of B. pumilus, and with the strain FO36B, we were successful. That is when Bob took the stage. In total, we have 5 images out of our 6 JMU phages. We believe that these 2 Bob phages may be in love. Gadir shows some more detailed striations of the tail and some ill-defined tail end structures. In addition to Bob and other family members that we will describe, there were 2 other clusters that included giant syphoviruses and tiny podoviruses. Shifty 23 and McLean have a contracted tail and a headless tail as seen in McLean. Their capsid diameter is approximately 82 nanometers. Their extended tail is about 160 nanometers, and their contracted tail is around 54 nanometers.
These phages were then partly annotated by several classes, being used as genomes for teaching annotation in preparation for analysis of more recently discovered and sequenced phages. Gradually, 6 Bob family members were analyzed and are now ready for the final annotation steps. The exception to this was Shifty 23, and that was the focus of our work this semester. This table shows a comparison between each of the phages isolated from JMU and the bottom 2 not being from JMU but found to be highly related. This is an image from a Famarator map. The top ruler represents the genome of Pinkman, and the bottom ruler corresponds to Shifty 23. The slanted lavender lines between indicate nucleotide similarity and show that there are more nucleotides in Pinkman than Shifty 23. In that region, there are 8 predicted proteins only present in Pinkman, and that genome is thus longer by that many nucleotides. Other insertions and deletions, relatively speaking, account for the size differences in the genomes. No larger insertions are consistent between the genomes that might indicate a major horizontal gene transfer event. With this Famarator lineup of all 8 related phages, you can spot differences in gene sizes and arrangement. We're going to show you 3 examples of interesting areas in genes that can be studied in detail in the future. In this diagram, the DNA helicase gene is shown in the royal blue rectangles. As you can note by scanning, there are differences between the family members. In one case, there appears to be an insertion of an entire domain in Marcel and Gadir, indicated by the black square, and the helicase is missing at this location in Bob and Pinkman, indicated by the black ovals. But the helicase in these genomes is present some thousands of bases away.
This Famarator diagram shows the DNA polymerase gene in green with brown function boxes. Some are full-length genes, which are shown in the rectangles, and some of these are split into 2 genes, which are shown in the ovals. Between those 2 genes is a smaller gene encoding for what is called a self-splicing intron, which is shown in the smallest circles. Bob, Pinkman, and Shifty 23 are all examples of a split DNA polymerase gene. The gene intervening in Bob is an H&H homing endonuclease, likely to be a part of a self-splicing intron. Domains matching the DNA polymerases and other bacillus phages are present in both genes that flank the self-splicing protein gene. More investigation is required to document these patterns, which are present in larger families of phages. The ribonuclease reductase subunit genes, which are the brownish genes that are shown in 2 or 3 pieces in Figate, Bob, Marcel, and Pinkman, are separated into 3 parts by the presence of an additional H&H endonuclease. The ribonuclease reductase functions as an enzyme that is involved in the synthesis of DNA and can convert RNA back into DNA. This division of the ribonuclease reductase gene is something we want to research further, as well as the previously mentioned sections where genes are inserted or are different sizes. We are confident that our semester's work will launch new insight about Shifty23 here at JMU and will launch us into our next project involving the Ray 17 family.